And thank you, Dr. Iwakuma. I'm honored to have a chance to speak in this Congress. And thank you for attending this session. Today, I will be focusing on two basic points. First, about the detailed mechanisms of bovine respiratory mucosal immunity, and second, relationship between the nasal mucosal vaccine and the bovine respiratory mucosal immunity. Let me begin by talking about the bovine respiratory diseases. First of all, as for direct damage, death and treatment cost is well known. Second, not only direct damage, BRD causes indirect damage too. As for indirect damage, growth defect and declines in meat and reduction in reproductive rate and reduced feed efficiency and etc. are known. From the above, BRD is recognized about the most common and costly diseases. And so, a prevention strategy of BRD is very important. Before thinking about the strategies, let me talk about the risk factor of BRD. First, there is infectious agents such as virus and bacteria that actually cause the BRD. And second, there is host characteristics such as immune status and genetics to prevent infection. And finally, there is environmental factors such as transportation, ventilation, and sale and effects that affects growth of infectious agents and host immune status. As for prevention strategies, control of infectious agents was in particular focused. So the research of the vaccination and metaphilic cases were actively carried out. So various new vaccine using migration antibody method and long-active antibacterial have been developed. By the way, is BRD actually decreasing due to progress of prevention research? And this is the case of our Kagoshima prefecture. Unfortunately, it can be seen that the number of cases have hardly changed, and the proportion is increasingly. Thus, BRD does not decrease, but rather increase in Japan. And so, why does not BRD decrease? I think there are 4 factors described below. First, there is pathogens without vaccine, such as Mycoplasma bovis. And second, new pathogens such as influenza D. Third, unavoidable feeding stress, such as transportation and sale. Finally, there is problem with drug-resistant bacteria. Well then, how to decrease the BRD? I think it is important that we know the correct information about enemy and ally. So what is essence of the BRD? Unlike human and horses, BRD progressed gradually in the bronchoalveolar region, and it caused tissue remodeling by chronic inflammation. So the essence of BRD is alveolar pneumonia. Thus, true enemy is pathogens actually infected at the alveolar region instead nodes. And ally is respiratory mucosal immunity instead peripheral. However, it is considered difficult to beat specific enemies for the reasons mentioned above. So we are paying particular attention to respiratory mucosal immunity. Here, let me talk about mucosal surface. And like these images, most of cattle diseases occur on the mucosal surface, but why is that? Animals are in contact with the outside world through the mucosal tissue covering the inside of the body as well as the skin. So mucosal surfaces are not merely responsible for digestion, respiration, and excretion, but the major infectious pathways of many pathogens. And therefore, more than half of the immune cells present in the body reside in the mucosal surface, such as intestinal tract, respiratory tract, and nasal cavity. And therefore, mucosal surface have the unique immune systems. And this is a schematic diagram of the most well-known Peyer's patch of intestinal tract immunity. It is well known that such lymphoid tissues are found on various mucosal surfaces. And the most characteristic feature of mucosal immunity is common mucosal immune systems. That lymphocytes matured on the mucosal surface migrate to effector mucosal cytes through the thoracic duct and blood circulation. So mucosal immunity is completed with mucosa, and in order to prevent the diseases occurring in the mucosa, it is necessary to rebuild the immunity in the mucosa. Studies on mucosal immunity are progressing in human and rodents, but can we exploit the findings directly to bovine? I think we can't. And that's because bovine have different immune system from human and rodents in various ways. For example, first, the proportion of lymphocytes is in peripheral blood is higher than that of neutrophils. Second, the ratio of gamma delta T cells in lymphocyte is 15 to 60%, about 1 to 5% in human and mice. And finally, T cells showing regulatory action found not in alpha beta chain CD4 positive cells, but in fraction of the gamma delta T cells. Thus, I think bovine immune system must be examined in bovine. So, in order to study respiratory mucosal immunity of bovine, we apply Bronchoalveolar Lavage, abbreviated BRD, using endoscope. As a detailed method, insert the endoscope from nostril and proceed while performing local anesthesia. And after reaching the bronchoalveolar region, injection normal saline and recover injected saline. Then, the obtained saline solution is called a bronchoalveolar lavage fluid, abbreviated as BALF. BALF recovered from healthy bovine is colorless and transparent. And BALF recovered from pneumonia is like this. So we studied the respiratory mucosal immunity of bovine using these BALF samples. And first, I will explain immune cells present in the bronchoalveolar region of a healthy cattle. The bronchoalveolar region of healthy cattle is such a pink color, and cells taken from here is like this. Almost all alveolar macrophages, such as mononuclear and wide-side plasm, and including vacuoles. So, the character of bovine alveolar macrophages were examined in detail as compared with monocyte-derived macrophages. First of all, we analyzed the surface antigen expression. As for alveolar macrophage surface antigen expression, like this. As for monocyte-derived macrophages, like this. So surface antigen expression was different. And especially, alveolar macrophage highly express CD21. CD21 is complement receptors that mainly express at B cells and follicular dendritic cells. On the other hand, it is reported that expression in macrophages is not observed in various macrophages, including alveolar macrophages in human and rodents. So, alveolar macrophage express CD21 is a unique property in bovine. Subsequently, we analyzed the phagocytic ability using fluorescent beads and flow cytometry. As a result, histogram of alveolar macrophage like this, and other monocyte-derived macrophage like this. When these results are represented by dot plot, it becomes like this. And alveolar macrophage has higher phagocytosis rates than monocyte-derived macrophages. So, you can say alveolar macrophages have spontaneously high phagocytic ability. Let me briefly summarize character of bovine alveolar macrophages. First, bovine alveolar macrophage uniquely express CD21, complement receptor 2. I predict that CD21 is involved in phagocytosis of opsonized antigen in bovine. Second, bovine alveolar macrophages have high phagocytic ability. I predict that alveolar macrophages have high phagocytic ability is they must remove not only pathogens but also nanoparticles invading with respiration. Let's move on now to the immune response in the bronchoalveolar region of cattle with respiratory diseases. When light inflammation occurs, infiltrate of excudent occur like this. And interestingly, infiltration of neutrophil is not observed in the early stage of infection. And the infiltration of lymphocyte alone is recognized like this. Thus, when the phagocytic antigen invades, it is thought that lymphocytes and alveolar macrophages cooperate to eliminate pathogens first. And next, when burst pathogens and pathogenic antigens that cannot be treated by macrophages and lymphocytes is invaded. Bronchial tree becomes red like this, and the inflammatory images become visible. Then, neutrophils will be recognized like this. In addition, when neutrophils infiltrating tissue cause necrosis, they scatter large amount of reactive oxygen species and proteolytic enzyme and destroy normal tissues. So, like this images, macrophage phagocytose apoptotic neutrophils and recognized. This is an important reaction in the healing process called efferocytosis. So for healing, apoptotic neutrophils must be removed by efferocytosis. So if infection cannot be controlled and persistent neutrophils infiltration occurs, a pus will stick to the bronchi like this. And the number of immune cells in birth will be 10 to 100 times. And most of the cells that have increased are thus neutrophils, and when they cannot be completely treated by effect of efferocytosis, fibrosis of lung tissue is occurred by reaction oxygen species and proteolytic enzymes. And this is CT images pneumonia case and whose fibrosis has started due to severe infections. And furthermore, as pulmonary fibrosis progresses with chronic infection, at first glance, the bacterial alveolar region appears to have no inflammation. But cells are hardly isolated from birth. So it is thought that this is a characteristic of bovines that is also found in another infectious diseases. The infection site is isolated by fibrosis. When this state is reached, as can be seen from these CT images, only about 20% of the lung lobe have normal function, and the prognosis will be poor. Let me briefly summarize changes in respiratory mucosal immunity by infections. Initially, in healthy cattle, the resulting cells are almost all alveolar macrophages, and they have high clearance ability. And next, when phagocytic antigen invasion, only lymphocyte infiltrates to the bronchoalveolar region, and lymphocytes and alveolar macrophage cooperate to eliminate pathogens. And next, when burst pathogenic antigen that cannot be treated by macrophages and lymphocytes is invaded, inflammation causes and neutrophils infiltrate to the bronchoalveolar region. And if it succeeds in elimination of the pathogen, apoptotic neutrophils removed by efferocytosis. Next, if infection cannot controlled and persistent neutrophil infiltrations occurs, fibrosis of lung tissue is occurred by neutrotic and reactive oxygen species and proteolytic enzyme. And lastly, pulmonary fibrosis progress with chronic infection, and the prognosis will be poor. So next, let's move on now to detailed function of alveolar macrophages, which are thought to play the most important role. Comparing the surface antigen expression of macrophages collected from healthy cattle and macrophages collected from cattle with pneumonia. As I mentioned earlier, macrophages from healthy cattle is expressed CD21 and not express MHC class 2. On the other hand, macrophages from cattle with pneumonia was downregulated CD21 expression and upregulated MHC class 2 expression. Thus, alveolar macrophage use power for phagocytosis when healthy, but when infection occurs, it is thought that power is used for antigen presentation. And by the way, what factors are controlling alveolar macrophages? We believe that hormonal factors, including cytokine, play an important role. So we conducted the following assay. We collected blood from healthy cattle and cattle with pneumonia and compared the concentration of various hormonal factors in the centrifuge supernatant by ELISA. This is the dot plot graph of the result. There is no significant changes in IL-2, IL-4, IgG, and IgA. But interferon gamma and IL-8 have a significant increase in pneumonia groups. And especially, interferon gamma can hardly be detected in the healthy groups, but there are individuals over 100 picogram per ml in the pneumonitis groups. So, we consider that interferon gamma is a key factor for regulating the function of alveolar macrophages and followed by the ex vivo assays. The test cattle was 1-month-old male Holstein, and the bulb was performed to isolate alveolar macrophages, and the isolated macrophages were cultured in control media or media containing recombinant bovine interferon gamma, and these assays were carried out. First, let's see the result of the phagocytosis assay. And we did it fluorescent bead assay as mentioned above, histogram of control culture like this, and when stimulating with interferon gamma is like this. And the result of each percent is as shown in this dot plot. So, it can be said that the phagocytic ability of the alveolar macrophage is decreased by interferon gamma stimulation. Next, let's see the result of surface antigen expression. Histogram of control culture like this, which is almost unchanged before culturing. But when stimulation with interferon gamma, the expression of CD21 is drastically decreased, and the expression of MHC class II was drastically increased. Thus, it was found that interferon gamma is a major regulator of changes of surface antigen expression of alveolar macrophages, and interferon gamma downregulated CD21 and upregulated MHC class II expression. And lastly, let's see the result of mRNA expression by real-time PCR. And that shows how many times increased by interferon gamma stimulation compared to the control culture. Thus, the TNF-alpha associated with inflammation hardly changed, but the expression of INOS related nitric oxide production and CCL5, a chemokine associated with infiltration of lymphocytes, was greatly increased by interferon gamma stimulation. Let me briefly summarize control of alveolar macrophages by interferon gamma. By interferon gamma stimulation, it downregulates phagocytic ability and CD21 expression, and upregulate MHC class II and mRNA expression of INOS and CCL5. These changes are similar to changes in alveolar macrophage due to infection, and it can be said that interferon gamma is a major factor that changes the function of alveolar macrophage from phagocytosis to lymphocyte breaching. Then, what does it mean alveolar macrophages change to breaching function? In order to think about this, we need to know about the paradigm shift concerning innate immunity in recent years. The biggest finding is the discovery of pattern recognition receptors, abbreviated as PRRS, including toll-like receptor that won the Nobel Prize in 2011. In the past, recognition of pathogens was thought to be work of T cells of adaptive immunity, but in fact, PRRS of innate immune cells recognize pathogens by pathogen-associated molecular pattern, abbreviated as PAMPs, and then starts the response of a series of immunity. So it can be said that advanced immunity, including the innate and adaptive, begin with the recognition of PRRS. And furthermore, it has become clear that macrophage plasticity. In the past, macrophages are thought to be cells that simply non-specifically phagocytize antigen and cause inflammation. However, recently, it has become clear that macrophages have various subsets as well as lymphocytes, and that subsets change with niches. In particular, macrophages also have a subset as well as regulatory T cells. So, suggesting that they are deeply involved in the regulation of fall immunity. From these findings, immunity has come to be recognized as a mechanism for maintaining homeostasis that keeps itself as self, and not merely to eliminate antigen. So, what does it mean to upregulate immune system? I think as follows. In the past, weak immune system is susceptible to infection, and when it is strong, it is less prone to infection. But it get too strong, it will be developed autoimmune diseases. But with the discovery of immune cells with suppressive ability, I believe that if immune system is weak, it is more prone to infection and allergy come out, and if the immunity is strong, it is neither infectious nor allergies. Let me briefly summarize paradigm shift of innate immunity. In the previous concept, pathogens recognition is a function of the T cells. So the role of innate immunity is said to be merely non-specific phagocytosis, and the regulation of immune function was thought to be done only by adaptive immunity. However, after discovery of the PRRS, it is clear that pathogens recognition equals start immune response begin with innate immunity. So it is recognized that the function of the bridging linking innate and adaptive immunity is more important. Thus, regulation of immune function is thought to be done by innate and adaptive immunity in cooperation. Next, let's think about upregulation of innate immunity. In the previous concept, upregulation of innate immunity means to improve merely phagocytosis activity and digestivity and pro-inflammation cytokine production. On the other hand, in the new concept, upregulation of innate immunity, including bridging to adaptive immunity, means to improve the ability to regulate the correct immune response to preserve homeostasis. Then, as to how such complicated adjustment is performed in innate immune cells, in recent years, the mechanisms that pumps cause epigenetic changes in innate immune cells, and it memorized. Then let's discuss about respiratory mucosal immune response in the bovine. Almost all immune cells in the bronchoalveolar region of healthy bovine are alveolar macrophages expressing CD21. And it phagocytizes non-specifically, invading foreign substances. Here, when an amount of pathogenic antigen that cannot be processed by only macrophages is invaded, PRRS of macrophages recognize pumps, and macrophages are activated. Furthermore, macrophages function changes in the bridging direction by stimulation of interferon gamma and express MSG class 2. And lymphocytes and macrophages cooperate to eliminate pathogenic antigens. Then let's move on the second topics, relationship between nasal mucosal vaccine and bovine respiratory mucosal immunity. Nasal mucosal vaccine approved in Japan is TSP2, which was released about 4 years ago. As a feature, it is a temperature-sensitive attenuated vaccine. Only bovine herpes virus 1 and parainfluenza virus type 3 are included as an antigen. But interestingly, since launch, there has been a voice from clinical practice that respiratory diseases other than BHB1 and PI3 also decreased. And I feel that there is also a therapeutic effect. Then, what is the such antigen non-specific response of TSP2? We focused on the fact that it is an attenuated vaccine administered to nasal cavity, and thought that TSP2 might regulate the function of innate immunity. In other words, as it is an attenuated vaccine, so TSP2 also have pumps and should stimulate PRRS such as alveolar regions. And then let's summarize the hypothesis. And TSP2 include viral pumps which stimulate PRRS of cells present in the respiratory tract, including alveolar macrophages, and is regulating mucosal innate immunity. In order to study about this hypothesis, we conducted the following research. As material and methods, the test cattle was 1-month-old male Holstein, and we administered TSP2 at the timing as shown in the figure, and the valve was carried out to collect valve. And the recovered valve was centrifuged and separated into supernatant and cellular components and analyzed for each component. First, in order for TSP2 to affect cells in the bronchoalveolar region, it is necessary to actually reach the bronchoalveolar region. From the nasal swab, viruses derived from TSP2 have been shown to be recovered from 1 to 60 days after administration, but the bronchoalveolar region is unknown. So we isolated total DNA from the supernatant and performed PCR using primer for BHB1. As a result, a positive band appeared on day 7 of administration. And in order to confirm whether this is really derived from TSP2, we performed a sequence of SNPs and SNP regions according to this paper. And we found that the virus detected from 7 days birth has the same SNPs as TSP2. From the above, it was revealed that TSP2 actually reached the bronchoalveolar region, and the pumps of TSP2 can influence PRRS of cells in the bronchoalveolar region. Next, we analyzed changes in the characteristics of alveolar macrophage recovered from cellular components. Regarding surface antigen expression, expression of CD21 decreased at 14 days, and the expression of MHC class II increased on 40 days after administration. And next, as for mRNA expression, expression of INOS and CCL5 was increased to 40 days after administration. Next, interferon gamma concentration in the humoral component as analyzed. It is increased on 40 days after administration. And furthermore, although this is still a preliminary reporting stage, and when cultured alveolar macrophages with various pumps and TSP2 in ex vivo, it is revealed that the saturation of H3K9 and methylation of H3K4, which is epigenetic changes of activate transcription, was also increased when stimulation by TSP2. Let me briefly summarize control of bovine respiratory immunity by TSP2. Administration of TSP2 upregulate expression of MHC class II, INOS and CCL5, and downregulate expression of CD21 of alveolar macrophages. In addition, the interferon gamma concentration is in the bronchoalveolar region also increased. This suggests that TSP2 can change to bridge the direction of bovine respiratory mucosal immunity and can induce to the correct immune response. And that we believe that this is done by epigenetic regulation by pumps contained in TSP2, and we are currently conducting further research. And finally, I'd like to recap what's been said so far using Conrad-Waddington epigenetic landscapes. And suppose certain pathogens invade, and it leads to lack of response of innate immune reaction. The pathogen will then proliferate in the host and cause serious infection. On the other hand, suppose that a certain pathogen infiltrate and induce excessive immunity, like inflammation and autoimmunity. As a result, it it induces fibrosis, and it becomes impossible for the immune response to proceed, which in turn leads to lack of response and causing proliferation of pathogens and cause serious infections. So, if TSP2 is administered in advance, TSP2 can guide respiratory mucosal immunity to the correct response. And then, even if various pathogens invade, they can be eliminated with the correct immune response. So, I'd like to end my presentation, but I'd like to express my thanks to everyone at the end. Thank you so much for your kind attention.
Thank you very much.